Hi everyone, thanks Kim for that. I have the distinct pleasure of introducing my mentee Yassmin. I've had a really great time working with her over the past 5 months, and she is so passionate and so curious, and I'm really excited for her to be able to share her project on genetic engineering with you. She came in with a very strong idea of wanting to work with biology, and in these difficult COVID times, it's been great to see her be able to build out that interest and really create a product that she can share with other people to share what she learned from her research. And I'm excited to see where she goes with it in the future, and can't wait to see her get into a lab and get her hands on some of the techniques that she's been learning about. So I'll let her take it from here.
Thank you, Michelle. So for a bit about my mentor, Michelle Dionne, she is employed in MIT and she's a PhD candidate there, and she works in medical physics and medical engineering. And she also does research in immunoengineering and biomaterials. And she is also originally from Lunenburg, Massachusetts. And in her free time, she enjoys reading, writing, going on runs, and baking. And for my project overview, I created a website for my final project on CRISPR-Cas9 research and gene editing. The website talks about the gene editing tool and many other things that revolve around CRISPR. I chose this project because I'm interested in genetic engineering and how we can alter our DNA. For my future work, I want to be a genetic engineer. And I was able to get as far as to publishing the website so that anyone can see it. So my magic experience was great, and I was able to spend a lot of time outside of school researching gene engineering because I'm interested in that. And my experience in the magic program was great because I was able to explore science and learn more about genetic engineering and medical engineering. So for my website, for my project, I created a website, and it covers many things like the overview of CRISPR, the benefits of it, the development, the evolution, the ethical background, the role of guide RNA, the significance of it, the meaning of Cas9, GMOs, gene drive, and mutations. And here is basically my website that I created about CRISPR, and it talks about CRISPR and many other things related to it. And moving forward to the overview, CRISPR is a gene editing tool that stands for clustered, regularly interspaced, short palindromic repeats. And the Cas9 enzyme is a protein complex that acts as a cutting scissor to break off segments of DNA. And scientists use this method to modify genes by altering a gene using either insertion or deletion. Moving down to benefits, this tool can be used to cure diseases such as HIV, herpes, AIDS, Down syndrome, and even cancer. This editing mechanism is also being used to develop COVID-19 vaccines. And moving forward to the development, there have been many historical accomplishments that this tool has provided us, such as modifying plant, animal, and bacterial cells. And in the 19th century, they used gene engineering to modify the mitochondria of a human embryo, resulting in the first human with 3 parents. And moving down to the evolution, mice are an example of an animal that scientists usually test on. Scientists discovered more clustered repeats in DNA. And also in 2002, the name CRISPR was published. And a lot of people remain skeptical about this tool, but it will always affect us. And moving down to the role of guide RNA, in DNA, the base pair adenine always pairs with thymine, and cytosine always pairs with guanine. And in some cases, there's an alteration that doesn't follow this process, but with a copy of guide RNA, it is able to lead the Cas9 enzyme and locate the place that needs to be modified. Moving forward to the significance of CRISPR, CRISPR is a cluster of DNA segments located in the genes of prokaryotic organisms, and it is used to break down DNA from similar bacteria. And moving forward to the overview of Cas9, it is a molecular tool that is used to detach areas of the DNA sequence, and it is made up of proteins and cuts the targeted area. And also moving forward to GMOs, many organisms such as mosquitoes, fleas, and other insects carry around a lot of diseases that are harmful to the human race and can even lead to death. CRISPR-Cas9 can be used to insert antibodies to these organisms so that they do not pass on this virus to humans and that they are immune to malaria. And moving on to part 2, Cosmo is a genetically engineered cow that was given an extra SRY gene to chromosome 17, and Cosmo was originally female and had the XX chromosome, but with inserting the SRY gene, the cow was able to have male characteristics and was more useful because they are more efficient for agricultural purposes and reduce the carbon footprint. And leading down to gene drive, it can be used to turn organisms who have a heterozygous gene into a homozygous gene so that they are able to express a certain trait. And by doing this, the animal will pass down its trait to further generations. And lastly, mutations. CRISPR-Cas9 is an amazing tool that is superior to the world, but with everything comes disadvantages, such as mutations. While editing the DNA sequence, you may awaken the wrong gene and target the wrong thing. Lastly, people disagree with the uses of this tool because people might fear that it would be used inefficiently to alter things in our ecosystems. Thank you.